developmental unit is in the intestine. If you look at the inside of the intestine, it almost looks like the fingers on your hand. You've got these little finger-like projections. They create the surface area to absorb food, and they're called villi. That's this part here. There's a part of the intestine that harbors very many stem cells, probably more stem cells than any other organ in the body taken together. And as these cells differentiate, they migrate upwards, and they can become goblet cells that make mucus. They can become endocrine cells that make hormones, and those are the ones presumably that become carcinoids, or they can become absorptive enterocytes. And then there's another group that can become Paneth cells that go the opposite direction as well. And this is a process that goes on and on and on. This process, they turn over probably 5 days. Every 5 days, you get a new lining to your intestine. So there's always an opportunity for these cells that are arising from the stem cells to acquire mutations and potentially turn into cancer cells. And this must almost certainly be what happens in carcinoid. Now, this is a similar slide to what Dr. Shiv Dasani talked about, which is what is generally believed to be the way that endocrine cells become specialized. Stem cells get committed to becoming secretory cells by a protein called MAF1, one gene. There's another gene called neurogenin-3 that somehow, if it's expressed at a high enough level, makes the secretory progenitor cell, in some cases, become an endocrine cell. And then these endocrine precursor cells express another related protein called neuro-D. This one is the one that seems to be important for the end part of differentiation. This may be the gene that makes cells stop dividing. And this will be important for what I'm going to talk about in the next few minutes, a little bit later down the road, that this one really occurs at the end of differentiation that helps ensure that cells stop dividing. We did a number of cell lineage tracing studies over the years to try and make ladders like Dr. Shiv Dasani showed you in the prior talk. And it turned out we almost all along the way, we found things we didn't expect to find. But for the most part, what we found is that this gene that's important for becoming an endocrine cell, NGN3, can even be expressed in stem cells. It can be expressed in these secretory progenitor cells, because we can follow what happens to them because these cells essentially have the blue marker. And then essentially almost all the endocrine cells seem to arise from this, the cells that make neurogenin-3. But then we got hit by a big surprise. About half of the serotonin cells did not. So this is presumably the cell that might give rise to carcinoids. And they become specialized by a totally different process that we never thought existed, and it's something that's an area of very active investigation that we've actually has been supported by the Caring for Carcinoid Foundation. We did a similar ladder type study looking at neuro-D. This is that protein that's important for making the cells stop dividing and become terminally differentiated. And what we found is, for the most part, when a cell makes neuro-D, it can only become an endocrine cell. But there was a problem. About half of the serotonin cells didn't come from neuro-D cells. And one of the real questions we're asking right now is, where do they come from? I think the reasons for doing this is that it's really hard to know how to understand what goes on in becoming a carcinoid tumor or to make an animal model for it, which there really isn't a good model for, without understanding what the cells are that give rise to it. And I think that one of the reasons we're interested in this cell here is it doesn't make neuro-D. That's the gene that normally makes the cell stop dividing. It could well be this is the cell that's going to be most susceptible to becoming a carcinoid tumor. And I'll show you a little bit about how we might go about trying to find that out in work that we've already done and work that we plan to do. What sort of got us started in this way was a paper that was published about 8 or 9 years ago in Japan that said a lot of carcinoid tumors have mutations in a genetic pathway called Wnt. Now, you don't really need to know the details of what Wnt is, except this is probably one of the most common pathways that's associated with very commonplace colorectal cancer. So it's really an important oncogenic pathway in humans for colorectal cancer, a very common type of gastrointestinal cancer. And it may be important in some carcinoids. This is not, it's a little bit of a controversial paper. So we thought that we might be able to try and take advantage of this observation to see if we could make an animal model. And for a lot of other reasons. So the idea here was, could we genetically engineer a mouse where we could activate this pathway in one of two cell types, an early endocrine precursor cell that made neurogenin-3, or at a relatively late stage in the process, a cell that makes neuro-D. And with these mice get neuroendocrine tumors. We had no idea what to expect, to be honest with you. But as it turned out, when we activated this pathway in the neurogenin 3, the early cells, here you see a section of the mouse small intestine that shows lots and lots of tumors. These are actually polyps. And as it turns out, they're probably not malignant for the most part. They don't develop metastasis like many human carcinoids do. They're pretty much limited to the intestine. They're not very invasive. This one, I think, is what we call carcinoma in situ. But again, we don't see a very high level of metastasis the way we do in humans. But we got cancer. So now the question is, what does this have to do with neuroendocrine cells? Well, when we stain some of these for serotonin, the major gene product in many carcinoids, you can see these polyps made lots of it. And this isn't a general feature of other types of intestinal cancer, because if we activate Wnt signaling in all intestinal cells, they really don't make very much serotonin. So it seems like this has resulted from targeting the activation of this Wnt signaling to an early endocrine precursor cell. And in fact, it may matter a whole lot what cell you targeted to in order to get a cancer, because I don't know that we necessarily got the right one. These are mostly adenomas. They're not adenocarcinomas. So they're what we would call large polyps. There's a lot of them here. So we've clearly done something. Now, when we did the same experiment with neuro-D, so if we activate the Wnt signaling in the later cells, the cells that are almost mature, unlike the ones I just showed you, where they get more and more polyps the older the mice get, and they die as the mice get older, these were happy, healthy mice. They didn't get polyps, and they lived to a ripe old age for a mouse, 2 years, with no cancer. So this tells me that if we target and activate Wnt signaling in a relatively mature precursor cell, that's not the right one to develop a model of carcinoid, because at that stage, you can't activate the pathway in the right way. And I think that's very important. And this is what comes back to this other group of serotonin cells I told you a few minutes ago. This is the sort of the standard pathway of differentiation, the ones that come from neurogenin 3 and neuro-D. What about the other ones, the ones that don't make neuro-D, that don't have neuro-D shutting off tumorigenesis? Do they have a greater malignant potential or a greater potential to become a better model for carcinoid? And that's one of the things we're working on now. I'll just show you a little bit about how we're doing it. We don't know the answer to the question yet. So I'm going to skip over this for now. So to do this, we wanted to make some genetically engineered mice where we could make the cells glow different colors, much of what Dr. Shivdasani talked about a little bit earlier. And we started out, we wanted to be able to make all serotonin cells glow a certain fluorescent color. So we used a gene that was specific for serotonin, the first enzyme in serotonin biosynthesis called tryptophan hydroxylase, which is the first step. This is how serotonin is made, to express a fluorescent protein in a transgenic mouse. And so this here was an example of a cyan fluorescent protein. It's kind of a turquoise color, where we have made a mouse that every single serotonin cell is this color when we look at it as a special type of microscope. We also wanted to make a way to decide which of these are the ones that come from neurogenin 3 and which are the ones we don't know anything about where they're coming from, because those are the ones we really want to study. So we made another mouse here that had all the neurogenin 3 cells and their descendants. They're now bright yellow. And depending on how we run our microscope, we can look at one or the other. And the question is there a way we could use this to isolate each of the different types of serotonin cells to try and figure out where do they come from? So what we did is we crossed those two lines of mice so that theoretically what we would have are some cells that are serotonin cells here, that they have the turquoise, and they also came from neurogenin 3. So they're both yellow and turquoise, or cyan. But this other new class of serotonin cells that I told you about would not express any of the yellow. They would only be turquoise. And the question is, can now we use that to isolate those cells? And that's actually relatively easy to do. There's an instrument called a cell sorter that can basically separate cells. If we take these out of the intestine, make a suspension, and we put them into this machine, we can separate these from these. And to show you this is how the machine works schematically. It's actually a fairly expensive and involved apparatus. You can activate a laser beam here, and it causes the cells of different colors to get deflected, so you can collect them and separate them from the ones that don't have any color or the color you're not interested in. And I think now the idea is that we figured out how to collect these cells. We want to now find out what are all the genes that each cell expresses. And from that information, hopefully we can learn how they are different and what are the critical genes that are responsible for making this new class of serotonin cells. Once we've done that, we hopefully can then see, are these any more susceptible, can we make to becoming neuroendocrine tumors? And can we use that to make a better model in mice? Again, in many types of cancer, having a good animal model has proven to be very useful for trying to identify new ways to treat tumors. And that's one of the long-range goals we hope will come out of this work down the road. I think at this point, I'll stop and I guess turn it over to the next speaker and answer questions at the end.
During the panel. Thanks very much, Dr. Leiter. I'd now like to introduce Dr. Michael Choi of the Massachusetts General Hospital. And I'd also like to point out, Dr. Choi is the recent recipient of the Mary Therese Hartzheim Award for Neuroendocrine Tumor Research. And this was an award that was co-funded by the Caring for Carcinoid Foundation and the Mary Therese Hartzheim Foundation.
Well, thank you so much. It's an honor to be here, and it's an honor to really follow really two of my mentors, Dr. Ramesh Shivdasani and Dr. Andy Leiter, who I actually spent a lot of time together. In fact, I spent about 5 years with Dr. Shivdasani before setting up my own lab just a few months ago. And with Dr. Leiter, I mean, we published together, so we all know each other very well. Thank you for inviting me. And today, I'm going to be talking to you about actually one particular type of tumor called gastronoma. It's a type of neuroendocrine tumor that happens to not too many people, but when you have it, it's very serious. And really, the talk's going to be two parts. I'm going to talk to you about really one story that I've sort of kind of finished, quote unquote, in Ramesh Shivdasani's lab when I was there. And then I'm going to talk to you about what I'm going to be doing and what I'm starting to do now since I have my own lab and doing independent research. So this is just a slide of gastronoma. And it's just a simple slide that shows on the left, actually, do we have a pointer here? Okay, here. So on the left, you see this cancer here, basically, gastronoma, sitting in the duodenum, or the small intestine. This is the sort of type of thing that you will see if a gastroenterologist like me were to do an upper endoscopy and just really look in, and you see this a tumor sitting there. Once we resect this tumor and do a simple staining under the microscope, and you can see that the tumor is sitting sort of underneath around the normal small intestine, the villi that you've been hearing about, which is normal. But you also see this cancer that's sitting and growing. So I think the basic premise that I want to really introduce is that if we can understand developmental biology, we can better understand cancer biology. And if we can better understand cancer biology, that will eventually lead to better diagnosis and hopefully therapy. So what I'm actually working on right now is really developmental aspect of that science, of actually normal cells. So
Dr. Choi, excuse me one moment. Could you please just define for our audience what a gastronoma is?
Yes. So gastronoma is a type of carcinoid tumor, neuroendocrine tumor, that occurs in one particular type of endocrine cells. And these cells are actually found in the stomach, actually. And this is sort of the type of picture that you've seen with Dr. Leiter and Dr. Shivdasani's talk. I think they were showing the intestine, but this is a similar sort of thing. It's a very complicated slide, but I just want to focus on this panel right here. You see a similar type of structure in the stomach as well. And in the stomach, instead of crypt, these things are called pits. It's just a different naming. But basically, you see different types of cells here. And towards the bottom of that pit, you see these endocrine cells. And this is where many of our, quote unquote, neuroendocrine cell tumors probably start, because that's where neuroendocrine cells live. So for example, here, you see these cells called D cells, or sometimes you see these cells called G cells. And these cells represent different hormonal cells that express things like gastrin. And the gastronoma that I just spoke to you about is really the cancer of the gastrin-producing cells, or we call them G cells. So So what is gastrin? Gastrin is a hormone that's made by G cells here at the sort of bottom portion of the stomach. And when it gets released, it goes into the bloodstream, and eventually it goes up and reaches the upper portion of the stomach, where it reaches certain cell types that eventually lead to expression of stomach acid. And this is something that we all have, and we need to digest our food. So eventually, for example, these cells called parietal cells receive signals directly from the G cell or through the gastrin, or through another cell type called ECL cells, and eventually, basically, it expresses acid to digest their food in the stomach. So how do these G cells actually get started? Now, by the way, I've cut out many of the slides, by the way, just to fit the interest of time. So the package that you have may not actually follow the slides, but they're all the same slides, and some are missing. So this is sort of a slide that you actually saw with Dr. Shivdasani's talk. He was showing you about the intestine. This is actually a similar slide that's related to the stomach. So in the stomach, just like the intestine, has stomach stem cells that sort of live in that pit. And what's really fascinating, I mean, this is really one of the reasons I'm actually in this business, because it's such a fascinating thing when you have a cell that can actually give rise to all the cell types in that organ. Now, in the stomach, for example, it has four broadly cell types: the mucus cells that produce mucus, parietal cells that I just told you about that produces acid, chief cells that produces an enzyme that's also important for digestion, and finally gastroendocrine cells, which is really why we're all here. I mean, these are, in many ways, probably the cell types that actually ultimately give rise to neuroendocrine tumors. And this particular one, you can't say what type it is, but for example, G cell would be one example. So what actually makes G cells? So when I was in Dr. Shivdasani's lab, this is one of the things that I did. I made a quote-unquote knockout mouse. And knockout mouse is very simply a type of mutant mouse where you can just take away and nullify one of the genes that you're interested in and see what you get. So if that gene is important for example, making G cells, you may not see any G cell in that mutant mouse. Well, when I first made that mouse, I had no idea what this will do. We just knew that this gene called NKX6.3 is expressed only in the stomach, basically. And I think Dr. Shivdasani went into a little bit about what you're going to see. But basically, when we first made the mouse looked normal. I mean, it looked fine. It was eating. It was doing its thing. But when we opened up its stomach and looked at what kind of cells are there, and this is really the panel that you want to see. So normal stomach would look like this, and the brown cells represent the G cells. But in the mutant mouse stomach, you see very few of these brown cells because they're not making any G cells. And if you were to count this, you see that in the normal mouse, you have many G cells. In the mutant mouse, you have very few G cells. So right there, we knew that G cell, or NKX6.3, is a very important gene for generating G cells. If you were to actually take these mice and just measure the gastrin level in their blood, for example, the normal mouse, which we call wild type, has very high or normal amount of gastrin in their blood, whereas the knockout mouse or the mutant mouse has very little. And a corollary to that is, what's the acidic level of the stomach of these mice? If you take normal mouse, it's the sort of the normal acidic stomach of 3.9. And here, the lower pH means it's more acidic. In the knockout, you have very elevated